All right, good. Lisa, you're right back there. You're over there.
We are live, everybody. Thank you for joining us. I apologize we're a few minutes after 8 o'clock. That happens sometimes. I had a flooded basement, but it's not flooded anymore. Now I have a new carpet and everything is fine. Welcome to the live broadcast of TWIS, where we will record all of the podcast, but it's going to be edited a bit for the podcast version. So thank you for joining us for all of the fun right now. You get all the things right now. And we're going to go live. You ready? Everyone ready?
Mm-hmm.
I'm ready.
Everyone's ready. So we are going live in... I'm a little out of focus, aren't I? I'll have to fix that in a minute. My IT department is slacking. In 3, 2... This is TWIS. This Week in Science, episode number 812, recorded on Wednesday, February 17th, 2021. Fungus February Fun. It's going to be great. I'm Dr. Kiki, and tonight on the show, we will fill your heads with spider legs, death, and fun guys. But first...
Disclaimer. As a cold snap grips much of Texas and millions are without power and temperatures drop below zero, some strange news is coming out of the fossil fuel industry, maybe? Via the Texas governor and Fox News, that wind turbines are to blame for power outages. Yes, the Green New Deal, so alive and well in Texas, is the reason for the power outages, according to Fox News and Governor Abbott. Few fun facts. Denmark gets 41% of its electricity from wind. Germany, 21%. Canada and Sweden have hundreds of dedicated wind farms generating power, and they all work in the cold weather, which is very common in those countries. State of Iowa, a little closer to home, 40% of its energy comes from wind, the most in the United States. Now, Western Iowa is seeing rolling blackouts from this winter cold snap as well, but that's because 50% of its gas-powered generators went offline. Texas, on the other hand, only gets 10% of its energy from wind and has had similar issues with generators freezing. Not the wind turbines, mind you, but the gas generators are failing. The point is that government officials pointing fingers out of political convenience while ignoring the real issues is so 2020. It does nothing to solve the actual problems because it ignores the actual cause. And that's just the sort of dangerous thinking that led us to bring you This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
Gotta unmute myself. And a good science to you too, Justin, Blair, and everyone out there. What an energizing dance intro this week. This is fun. I love it. Welcome, everyone, to another episode of This Week in Science. We are back again. We've got all sorts of science to fill our show tonight. Also an interview talking about fun guys and genetics, genomics, all the good stuff. And I have stories about fish because fish are cool. I also have a story about, Planet 9. We got to keep up with Planet 9 and how less is actually more in your brain. Bum, bum, bum. Justin, what is in your segment of the show?
I've got killer caterpillars, Neanderthal resistance, and why snip calling is trickier than you thought.
All right, snip calling? Mm-hmm. All right. Mm-hmm. Okay, I want to find out all about that. Blair, what is in the animal corner?
Oh, I have cats, dogs, cockroaches, and spiders.
All the animals.
Yeah.
Yay. Cats and dogs. Hopefully there will be no fighting like or raining like. No, anyway.
No.
No. All right.
Oh,
So
somebody heard you.
Sadie heard me. We are joined by Sadie as well. It's wonderful. Jumping into the show, I would like to remind you that if you have not yet subscribed to This Week in Science, you can find us on Facebook, on YouTube, on Instagram, although we're not live videoing on Instagram, but we're there. We're on Twitter. We're on Twitch live streaming. And we are just about all places podcasts are found. Search for This Week in Science. You can also go to twis.org, our website, to find us. All right. Jumping hot off the presses, I want to talk about how we all need a little perseverance. You get it? Because Perseverance, the NASA Mars rover, is landing on Mars tomorrow. Less than 24 hours until it begins its descent of terror. And we can say that because the Curiosity rover had to go through its 7 minutes of terror or whatever it was. And they're basically using the same landing descent technique that the Curiosity rover had used previously. However, this time, as we've talked about before on this show, in interviews and stories, the Perseverance rover is not alone. It is carrying with it a little Martian helicopter called Ingenuity. And Ingenuity is supposed to test how well we can create aircraft for the Martian atmosphere. So that's actually a very interesting question because lower atmospheric density, but also a little less gravity. So does it all balance out? Or this is interesting physics at work here. Yeah, I had a great conversation with my 10-year-old Kai about the physics involved in aircraft. Beyond that, though, the Perseverance rover is going to be, once it lands, it's got a little tool for digging. It's going to be taking cores several inches down below the surface, below the regolith. And those cores, remember the interview with Mapperwocky? And he talked about how the Perseverance rover is going to dig up little cores and then leave them like little packages, little poops on the ground behind it. Mm-hmm. And then someday, another helicopter based off of Ingenuity, another craft will come around and scoop all those little collected bits up.
Scoop all the poop.
Scoop the poop. But it's not poop. It's just Martian soil. And take it and eventually have a return mission to Earth to bring soil samples back to our planet, which is really exciting. This is the long game they're working on right now.
See, this is important because this will give us our baseline before we all go to Mars and ruin whatever's happening.
I hope that there's no ruining. Yeah. I mean, there's Red Mars, Green Mars, Blue Mars by Kim Stanley Robinson kind of got people thinking in the direction of terraforming and, could we take Mars and turn it into something else that we want it to be, something more Earth-like. And another instrument on Perseverance called MOXIE is going to be testing conversion of carbon dioxide in the Martian atmosphere into oxygen. It's like a battery. It's going to use stored up energy. It's going to electrically, chemically convert CO2 to O2 to see if one day we can create oxygen that's breathable or oxygen liquid fuel that could be used to power aircraft. Then beyond that, there's a whole other study that came out this week that was really pretty neat, where researchers published a proof of concept using cyanobacteria from Earth in a simulated Martian environment with simulated Martian soil using everything in the proportions that they would be found on Mars: atmospheric pressure, temperature, molecular components of the atmosphere and the soil. And they found that nitrogen-fixing, oxygen-producing cyanobacteria can grow. Mm-hmm. Yeah, so maybe one day, this is also, all right, maybe there is life on Mars that we haven't discovered yet, or maybe one day we could take life there if we haven't already.
Yeah, Mars-infest destiny. That's the plan eventually. If there's nothing there at first, and then if there is something there at first, it's going to be so dynamically interesting to study that it'll have to pause the Mars-infest destiny for a little while, and then we'll go there, and it won't matter because we're going to need a second planet. We need a backup.
I don't want a backup planet. I
Yes,
don't
you
want.
do. Yes, you do. If you've ever had a hard drive crash that wasn't backed up, you immediately go, "Oh, I should have had a backup."
Oh, I know. Yeah.
I'm not saying I'd go.
Prudent to have a backup.
Yeah,
It's
gotta have
prudent.
one.
Yeah, it's good practice.
You
We
can
just got.
have Matt Damon and have him get the show started, and and some cyanobacteria.
That's right. And potatoes.
But yeah, still we got there's also like like you said, temperature. I think you listed it all. Temperature, pressure, the water,
The chemical
oxygen, and radiation is still going to be a thing for that's also one. So a little bit of work.
That's a
Long-term
tricky
thinking
one.
though.
That's the long game, everyone. We have to keep thinking long term. All right, long term. I'm just talking about right now. What story do you have, Justin?
Oh, me. Oh gosh.
Yeah, it's your turn.
Yeah. Okay, here we go. If you want to get away with murder, one of the first rules is what?
You tell me. I don't know.
Hide the
Have
evidence.
you never gotten away with the murder? No, yeah. Don't get caught.
Don't get caught.
Yeah. Don't let anyone know that you've committed a murder. Okay, so the tomato fruitworm caterpillars are doing just that. They silence their victims' cries for help as they devour their appendages. Of course,
Good.
these appendages are leaves.
Are they noodly appendages? No.
The caterpillars are eating plants. So how do you silence a plant from telling other plants that there's a predator there? Normally a plant does what? It releases a chemical compound into the air, right? And
Volatile compounds, right? Or compounds into the ground. But yeah.
Yeah, the volatile compounds. Exactly what we're looking for here.
Okay.
So those go out and they either attract a predator or they tell all the other plants to start some sort of other defense mechanism within them that might be a bitter taste or something of this nature. But what they found was that this is a team of researchers who have discovered that the caterpillars are releasing an enzyme that basically keeps the plant from expressing any of those volatile compounds into the air. This is Gary Felton, professor and head of the Department of Entomology at Penn State. We have discovered a new strategy whereby an insect uses saliva to inhibit the release of airborne plant defenses through direct manipulation of plant stomata. Stomata are the tiny pores. That's the way the plant gets those what are actually herbivore-induced plant volatiles, hip bees, into the air to either attract the predator of their predator or to alert the other plants to get their defenses up. It's also involved in CO2 exchange. So they've discovered it's making this very specific enzyme that it's in its spit that keeps the stomata closed, prevents it from interacting with the environment.
That's amazing.
It's like it's gagged.
Like,
Yeah, no, it's like
little plant. Nope.
and then it goes about eating it.
Yikes. Savage.
So it's a pretty interesting story, just as a one-off of the sort of arms race in nature kind of a thing. But they also think that they may be able to utilize this enzyme in the future if they need to keep plants from reacting to the stresses of, say, global warming or this sort of thing. It may be another tool or mechanism to be used in agriculture at some point.
Yeah, I wonder.
Potatoes on Mars.
Maybe it would help on Mars, right? You're not feeling pain from being in this
Everything's
habitat.
fine. Everything's fine.
It's fine. You're okay. Little potatoes can't scream to tell the other potatoes.
In space, when a potato screams.
Oh my goodness.
Yikes.
Yeah, yikes. Oh my goodness. I feel bad for the plants, but at the same time, it's amazing that these caterpillars are able to do that. I want to know more about more caterpillars. How many caterpillars have this ability? How many other insects have this ability?
Yeah, it's the first time they've seen one at all. So it's a pretty cool discovery by itself. It's also I feel like it wasn't that long ago. Wasn't even 10 years ago that we were first talking about these defense mechanisms of plants. I don't feel like we've even had that on the radar that long. And now finding, of course, there's countermeasures and a whole natural world arms race that continues to go on and on.
Of course there is. Who else has arms races? Blair?
Well, besides caterpillars, how about just cats? They have an arms race amongst wildlife. The cats that are allowed to go outside of homes have huge problems on wildlife, birds, lizards, small mammals. They're all just in big trouble from these savage predators. And a new study from the University of Exeter tells us that actually domestic cats hunt less wildlife if they are fed a meat-rich diet and played with more often. They were introduced to premium commercial food where protein came from meat as opposed to other sources, and that reduced the number of prey animals brought home by 36%. 5 to 10 minutes of daily play, which you have a cat and you're not playing with it for 5 to 10 minutes? That makes me
At
really
least
sad.
5 to 10 minutes. How could you have a pet and not play with it? Come on.
But anyway, all judgments aside, 5 to 10 minutes of play with a cat also results in a 25% reduction of wild hunting. So just two very simple things you can do there. Feed your cat a protein-based diet that's actually meat-based, which is good for them anyway, since they're carnivores. But also then interacting with them and playing with them. The play in this study actually involved simulating hunting specifically by moving a feather toy on a string on a wand. These are my favorite games to play with a cat. So that they could stalk, chase, and pounce. And then they would give the cats a toy mouse when they succeeded on pouncing so that they succeeded in their hunt. And so that would mimic a real kill. And so all of that to say, other measures like this bird safe collars, that reduces 42% of hunting of birds, but has no effect on mammals. And cat bills actually have no proven statistical effect on hunting, which I was surprised to hear, because a bunch of cats actually learn how to hunt
more quietly.
Despite the bell. Yes. Yes. So all that to say, forget it all. Just have an indoor cat. All of it to say, just have an indoor cat. But also, hey, if for some reason you have an older cat who can't adjust to their new way, if you get a new cat, please make it an indoor cat. But if you have an older cat that won't adjust to being inside all the time, play with it. Make sure it has a protein-rich, meat-based diet. Maybe that will help reduce this a little bit.
Now I'm thinking that the neighborhood cats that stalk around my house, I just need to leave plates of meat out for them so they won't kill the birds in my yard.
And play with them.
Some sort of
for 10 minutes.
That's right.
This sounds like a
I'll do
protection racket. Like the cats are like, "Hey, you like birds in your backyard? You don't want them to get hurt? Here's what you do: you leave out a little piece of meat for me every other day. You'll be fine."
Sure would be a shame if something happened to these birds.
And if you do that, then the cats win.
Yeah.
Yeah, no,
right.
Don't go against
They've
the
already
cat
won.
mafia.
It's
Don't
a
go
win-win
against-
for them.
I don't. They've got the
There's
weather
no such
racket,
thing
they've
as
got
La
the
Casa
food-
Nostra.
Cats and okay. Now we're just going downhill. How about we take let's take a trip. Let's take a trip away from the cats, Remember last year or a couple of years ago, we spoke with a couple of astrophysicists about their ideas, Mike Brown and Battigan. I'm forgetting his first name, Dr. Battigan, about their assessment of trans-Neptunian objects that are way out past Neptune and have these big elliptical weirdo orbits.
Yeah, one of the guys killed Pluto.
Yes, that was Mike Brown. Yes.
And now,
Yes.
because I think what his daughter told him that he like owes the galaxy, the solar system, a planet. That he's been now trying to track down
Planet 9. Planet
Yeah.
9. So Planet 9 is this hypothesized really big planet that's dark, that's way out there, past Neptune, past Pluto, just way out there. It's out in the dark, but it has a gravitational effect. And so the reason they think that there is a Planet 9 is because a bunch of these objects, according to their analyses that are out there, are clumping. And they say the clumpiness as a result of this big gravitational object that's way out there that's kind of herding these trans-Neptunian objects into clumps, kind of where they're associated. Well, a new study that is out this week that has been published by another group of astrophysicists, they have taken a look at a different group of trans-Neptunian objects, and they conclude that you can't conclude anything about clumpiness of trans-Neptunian objects because it's all about bias of where you're pointing your telescope in the sky and when. So there happens to be a bias to when we point telescopes to certain places in the sky because objects could be highlighted against the light of the Milky Way, or there could be other aspects to why they're looking in certain places at certain times. And so what they say is that they could not, I guess, get rid of the null hypothesis that there was no clumpiness. And so this clustering is something that is just a bias based, and there's no Planet 9. But Battigan and Brown are saying that, look, you just got to look some more. The clustering, it's there, and we're going to keep looking at objects, and you got to look at it in the right way, and there's going to be more telescopes, and we're going to keep looking. So different teams of researchers with different results who are scientifically battling it out for a planet's existence in the distant solar system. This is like reality television material, except not, because it's just science. It's reality, and we are working through hypotheses right now, and it's fabulous.
This is also why, like, everyone in college who got really frustrated at having to take stats, this is exactly why you have to take stats, is to figure out exactly how you can know if you think there's a thing. And if there is questionable doubt of whether there is a thing.
Yep.
There's a lot of amorphous words for saying, like,
I love the null
hypothesis.
The null hypothesis says there is not a thing.
Yeah, there's nothing.
There's not that thing.
You're tripping.
And then your
basically,
alternative.
it's the statistical version of you're tripping.
You're tripping, boo. Cold tripping. All right. Anyway, moving forwards. Oh, Blair. You had cats. Now do you want to talk about dogs?
Yeah, real quick, just a very silly study about dogs, and everything you assume about dogs is wrong. So your pet dogs, they're putting on a show for you. They are far more likely to play with one another when their owner is present and being attentive than when they are not. So they took 10 pairs of pet dogs that had lived together for at least 6 months. These are dogs that live together and play together all the time. They get along well. They ordinarily engage in play at least once a day. And then they videotaped these dogs under 3 conditions. 1, the owner was absent. 2, the owner was present but ignoring them. And 3, the owner was present and showering them with attention in the form of verbal praise and petting. And that overall, the availability of owner attention facilitated play. So if you were there and you were paying attention to your dogs, they were more likely to play with each other. They saw more bowing, hip nudges, wrestling, chasing, gentle bites, all the good dog playing stuff. So they have a few theories why this could be. 1, the owner's attention is a reward, and they're like young children asking their owner to watch them do something cool. Come see what I can do. 2, they might
Look at me. I can do a handstand. Look at me.
They think it's cute when we do this. We should do it. 2, they've learned that playing amongst themselves leads to bigger prizes, like the owner joining in or taking them outside. Like, you guys are getting too rowdy. Go outside. That's praise, right? That's reward.
It's
3,
reward.
they might actually provide a sense of safety, because even though they know each other, when dogs are playing, there's always a chance it could escalate and get a little nippy. And having the human there to intercede is like insurance. But 4, and this is the one I really like, is that the human's presence might be a trigger that creates oxytocin, which leads to an emotional state that manifests as play.
Interesting.
Yeah.
So it's the hormone neurochemical influence that leads to these behaviors, which are social in nature, the play and the sociality and the bonding that come from it. Pro-social behavior at the result of neurotransmitter hormones. Yes.
Yeah. So more studies needed, obviously, and this was only 10 pairs of dogs. It's not a huge sample size. But it is very interesting that you would think that, I would think 2 dogs that live with each other, if you left them alone, they would play all day. But it sounds like they're more encouraged to play when the humans are around, which is very interesting.
Nice.
Yeah.
Nope, they're not just keeping each other company. They're keeping you entertained.
Yeah.
So my last study, Justin, you don't have another story in the rundown. So
I've got 2 more, but I didn't.
Just for your
didn't
segment.
See another one there. Yeah.
Did you have one more for this roundup?
No, I didn't see a spot for it, so I didn't.
It's
no, there it is. I got a couple stories later. I want to jump into the interview.
Okay, awesome. All right, so my last story for this early part of the show, our stories to start things out, has to do with genetic diversity of salmon and trout and how we find out about that diversity where the fish are. You can catch the fish, or now there is environmental DNA. So you don't actually have to catch the fish. You just sample the water and get leftovers of the fish. You can find their DNA in little water samples or in soil samples from the bottom of a river or a stream, a waterway. Researchers at Oregon State University have just published their research looking at a bunch of different trout and salmon species in Oregon, northern and southern Oregon. And they found a lot of interesting connections between watersheds in northern and southern Oregon that are not connected. So like the Deschutes waterway and the Klamath waterway, they're not connected. However, people hypothesized that they were connected at one point in time, and they actually found genetic evidence from this environmental DNA that connects these completely distant subspecies, these different not subspecies, but these different populations of salmon, Deschutes salmon and Klamath salmon. They got some history in there, according to environmental DNA. They also were looking at another area. Let me see if I can find the Umpqua watershed area. They found a lot of really interesting stuff going on in Umpqua, where they found unique genetic variants of cutthroat trout, to a lesser extent, the rainbow trout. There was just a lot of genetic diversity that was not really expected. And so it is pointing a direction for researchers to look at the Umpqua watershed region for future investigation of these various species. Very fun stuff. But yeah, they went out, they sampled the environment, and they found these amazing connections between different fish species that they wouldn't have necessarily known about. Very cool. All right, everybody. This is This Week in Science. We are here, as we are every Wednesday, talking about science with you, and we hope that you are enjoying the show. If you would like some cool piece of TWIS merchandise, we have a store. Head over to twis.org, click on our Zazzle link, and you'll be able to find a veritable cornucopia of things that are TWIS in nature. We've got hats, we've got mouse pads, we've got mugs, we've got lots of items with Blair's Animal Corner art on them. So head over to twis.org, click on that Zazzle link, support the show, and enjoy some Twissy merchandise. Now I'll come back in and let me introduce our guest for the evening. Tonight we are joined by Dr. Ivan Liachko. He is the founder and CSO, Chief Science Officer of Phase Genomics, and one of the inventors of their core technology, which is called Hi-C technology. It's applying proximity ligation data to different facets of genomics. He received his bachelor's degree at Brandeis University in 2001, PhD in 2007 at Cornell, and then worked as a postdoctoral professional at the Department of Genome Sciences at the University of Washington in Seattle. He has authored more than 40 academic papers and a number of patents in the field of genomics and synthetic biology. He founded Phase Genomics in 2015 and leads the organization in an executive as well as technical role. Ivan, thank you so much for joining us tonight on the show.
Thank you so much for having me. This is super fun.
I hope that you're enjoying yourself. We try to enjoy ourselves and the science and all of it. We think it's fun, it's interesting, and if we can spark a little curiosity, that's what we try to do. And so I'm curious about your pathway into starting a business and entering into the field of genomics. How did you get interested in this field and end up where you are?
Sure, thanks. Yeah, I can give you a little bit of the origin story. So we
as long as there's superhero powers involved, like origin stories. Yes.
It could be villain. You don't know.
Oh no.
I'm an immigrant. I immigrated to the U.S. When I was 11 years old. I've always wanted to do biology. Always wanted to do genetics, and so I sort of started early on in life. I got my first job in a lab at the age of 16, and just kind of like pipetting stuff and doing like DNA jockeying. This was back in Philadelphia, and then like more of that in undergrad, more of that in grad school, postdoc, basically just like 20 years of of genetics. Then you get there and you're like, I don't have any other skills. I only know how to do research.
So I'll keep doing it.
Yeah, and so we did lots of cool stuff, lots of technology development during my years. And kind of towards the end of my postdoc, we came up with some cool technology. And I should say that, so there is a technology that you mentioned called Hi-C, which we use. We did not invent it. We just invented a couple of cool things to do with it, which is a distinction that's totally not important to you guys, but I'm going to go back and hear some stuff.
Very important.
I did not invent Hi-C. It was invented in Yuck lab at UMass by Eris Lieberman Aiden. But we've come up with some creative ways of using it that were new at the time. And we started doing all these crazy projects. And I'll kind of talk a little bit about them, what we do. But they were just very kind of just novel. And they went in different directions that were not originally intended. And they were working extremely well. And at some point, we were like, this technology is very powerful, is very useful to people all over the scientific community. But it's also like esoteric and complicated and kind of hard to use. If we don't turn it into a product, it's going to like fizzle out. Like it's basically like no one's ever going to hear about it. And that's true for so much of research, right? So like, when you work in academic labs, people are brilliant. They come up with amazing stuff all the time. But, as soon as you kind of like invent a thing, you just go on and you try to invent some other thing. And they focus on kind of like making it work super well once or twice, a couple of times. You publish it, you get it out there. Then you hope somebody else picks it up and turns it into something that can be used a lot. But it often doesn't happen. And so we were like, we don't want this technology to go away as soon as we stop working on it. And so we're like, we have to turn it into a product. Like we have to make it so that other scientists have access to it, that it's easy, that, it's something people can just use. It's applicable. And that's basically how Phase Genomics got started. And my co-founder, we knew each other for a long time. He was my DM in Dungeons and Dragons in my house many years.
That's awesome.
And so he left Microsoft, and which is where he was working for a long time, and we paired up and we started this startup, and we've been growing it since then. We got all this cool science going on. Yeah.
So what are you doing with this technology? You're making these things easier. So we talk about this stuff on the show all the time, but for maybe somebody who might be tuning in for the first time, what is the difference between genetics and genomics?
Sure.
The omic aspect.
So in one word, the difference between genetics and genomics is scale. So genetics, you're studying a gene or some genes. Genomics is when you're studying all the genes at the same time. So genomics is basically a combination of genetics and computer science. So it's basically DNA sequencing used to exist, and it existed before like the genomics era, where you would sequence like one gene, you would sequence like a thing. And now the new technology that is out, it's called next generation sequencing. It's when you're sequencing the whole human genome. You're sequencing the whole something. You're sequencing, you scoop a bunch of water from a pond with fish in it, and you sequence all the DNA, and you use computers to figure out where do these sequences come from, what are the different fish, etc. And so that's sort of the thing. And so it's sort of like genetics plus data, genetics plus, it's sort of like digitizing genetics. That's kind of what genomics is.
So you're a bioinformagician.
Bioinformagician?
Yes, right?
That is part of what I do.
Okay.
I've done some of that, yeah.
Okay.
I've done 20 years of like pipetting stuff. And then
so you've wet lab too.
Bioinformatics.
And now you're doing the bioinformatics stuff as well, and that's probably what's underlining a lot of what the company is doing.
Is both. So in our case, both. What's cool about this technology is that, and why we started the company, is because it's not just like you have like a new protocol for some wet lab magic. It's not just like a new algorithm for analyzing data. It's both, and you have to basically make both of them work together. And no one's going to do just one and not the other. And that was kind of one of the reasons why we kind of really felt like it would go away if we didn't commercialize it.
So I feel like this is the Shark Tank, and I don't know what the product is yet.
Perfect.
Because the first thing is like in anything in researching, when you've got a whole genome out there, where do you start asking a question or trying to narrow down so you can actually learn something from your data? Is one issue, and is that what Phase Genomics is sort of zeroed in on?
So basically, the high level summary of what we do is, what our platform does is it makes it so that scientists can discover more stuff from a lot of different genomic. To answer your specific question, like how does it start? Usually, the best way of going about something is having the question first.
Yes.
And then generating the data later, even though the opposite happens most of the time. Usually you're like, what a bunch of data, now what do I do with it?
Yeah.
But having, and then you pretend like you had a question the whole time. That's
Post hoc analysis.
That's right. Yeah. Like if they sequenced that fish pond and they discovered that it was full of like, I don't know, like virus that causes some disease, they'd be like, oh, you know what? We were studying this disease virus the whole time. You don't know what's out there. And that's kind of, that kind of is a segue to what I'm going to where I'm going to go with this. But what our tech does is it allows you to discover stuff in a lot of the sequencing efforts that you can't any other way. And so it applies to almost everything. It applies to doing things in your genome. It applies to doing things in the environment. It applies to agriculture. It applies to microbial stuff like microbiome and infectious disease, these kinds of applications. And so it's a sort of a hammer that hits many nails, and we have a lot of different applications for it. And so that's again, I still haven't told you what the product is.
Right. Because I've I've got over here, there's the next gen sequencing methods that are out there. There's the Illumina, there's the Oxford Nanopore stuff that can get these long reads or these very good small reads. And then you have the bioinformaticians creating the algorithms to decipher all of that information that comes out of it. So what is yours doing that's a step different from running it through the machine and running it through some algorithms that are already...
Sure. So here's how all DNA sequencing works. You take a DNA molecule, it's like a long string of letters, right? And you stretch it out, and you break it into a bunch of pieces, and you read the pieces, and then you have you have multiple copies of the same thing, and so that the pieces, when you break them, they're gonna overlap. You're gonna have staggered ones, and when you read them all, you can then try to overlap them back together to try to reconstruct what the original super long sequence was.
So
right?
It's a little bit better than if you cut up a book into small pieces. That's kind of tough to put all the pieces together. If you cut up 40 copies of the same book,
right,
or the same page of the book, then it becomes a little easier. Yeah.
Right. But here's the thing. So let's keep going with the book. You're right. You took 100 copies of the same book, you shredded them to tiny pieces. Now you try to reconstruct the book. You can do it. But here's the thing. What happens when you have a break between chapters? You have that blank page, right? Right. What happens when you have the same phrase repeated a bunch of different times? What happens if, I don't know,
repeats.
Right. So some combination of words. I was thinking of something funny, but I can't think of one. But it was the best of time. I don't know.
Right. If it said it over and over again for a page.
How do you know
anything with
on the other side of that, right? So that's the problem. The problem is that you're trying to take a sequence that's maybe 100 million letters long, or whatever, 100,000 letters long, 1 million letters long, super long, and there's repeats all over, and there's other sequences that are just hard. What happens if you have 100 A's in a row, right? Stuff like that. And so when you do these overlapping things, it never works perfectly. There's three main technologies that are used for sequencing DNA. One is from a company called Illumina. When they read the little DNA pieces, they give you very accurate reads, but they're short. They're a couple of hundred base pairs long, but they're very accurate. But you can only read 100 letters, like 300 letters at a time. There's another company called Pacific Biosciences, PacBio. They read 10,000 letters long, but it's super accurate, just like the Illumina ones. The difference is the Illumina ones gives you a ton of reads, hundreds of millions of these things. The Pacific Biosciences ones are longer,
but fewer.
Yeah,
less coverage,
more expensive. Right, coverage. Right, coverage is when you throw these little reads on top of a sequence. How deeply do they pile up? Right. Yeah. So super long, super accurate. Then you have Oxford Nanopore, which sequences even longer molecules, but they tend to be a little bit more error prone. And so there are these trade-offs, and they're also tend to be more expensive and stuff like that. But also in order to sequence a super long molecule, you need to actually purify a super long DNA molecule. DNA likes to break because it's a little. But no matter which technology you use, when you try to put that giant book jigsaw puzzle of sequence back together, it's going to be in chunks, in pieces, right? So you're going to get half a page here, a page there, and you're going to be well, how do they all fit together? What's chapter 1? What's chapter 2? Right? And you don't know. And so actually putting the genome together into actual chromosomes is extremely difficult. You can sequence, sequencing is easy, everybody does it, but actually putting it into an actual chromosome is really hard. And now
that's where your technology comes in.
That's where our technology comes in.
Yeah.
But let's add another layer to it. Let's say you're not sequencing just one person. Let's say you're sequencing a scoop of soil that has 10,000 different bacterial genomes in it, or some water with fish DNA in it that comes from God knows how many fish. And what else is in there? Algae, viruses, right? All this stuff. Now it's taking 100 books, 1,000 books, different ones, and throwing them together, ripping them up. Now you don't even know which pieces belong to which book, right? So there is this like sequencing DNA is fine, it's cool, it's totally common. Putting it all together is super hard. And so what our technology does, oh, thank you, I see. Our technology puts the jigsaw puzzles together. Long story short, here's a little video of how it works. So Hi-C technology was really neat. It was designed originally to look at how a genome folds together. That's what the original inventors invented it for, because when your genome is inside of a cell, it's like a hairball, and there's a lot of biology in there. It's like there's pieces of DNA that are touching other pieces, and they're folding around, and they're doing stuff, and it, like genes get turned on and turned off based on where they are. And this method, what it does is it captures sequences that are next to each other. And there's a bunch of molecular trickery that allows you to extract these little junctions where they're touching and sequence the junctions. And you can tell what's touching what. Now you would say, okay, I can tell like what's touching what. Ooh, this cartoon is great. I haven't watched this cartoon in like forever, so it's fun to watch it. So you basically you chop the stuff up, you glue the little pieces together, and then you can sequence. You're sequencing these junctions. It gives you a readout of sequence A and sequence B were connected. They must have been touching each other, and you're doing that for millions of sequences at the same time, right? Like bazillions of them all at once. And that essentially lets you count how often every sequence was touching every other sequence, right? And if you know which sequences are touching each other more, that means they were closer, and which sequences are touching each other less means they're further.
Right.
That's how you figure out the three-dimensional shape of of your genome, right? That's how awesome it is. It's like art. It's like using sequence to infer like three-dimensional stuff, right? And that was actually one of the reasons I really kind of fell in love with the technology, even before this company stuff we did. But here's the thing. DNA is basically like a long noodle, and so if you're close together, if you're touching a lot, that means you're close. If you're touching less, means you're far. Like
right.
In other words, three-dimensional distance and two-dimensional distance are related. So if two things are close to each other in 3 dimensions, they're also close to each other in 2 dimensions. So now imagine you have this jigsaw puzzle or book that's been ripped into pieces, but you know the 3-dimensional distance between every 2 pieces. Now you can put that.
You can begin to construct it. Yeah.
And this is where the bioinformagician stuff really comes into play, because it's not just 2-dimensional. You're starting to really put things into perspective.
You know, when we started that, just the wet lab part of the protocol, just doing the like laboratory stuff without even sequencing, took like a week, and it cost like $1,000 just to do one, just to do like the pipetting stuff, and you have to sequence it. It's another like couple grand, and then nobody knows what to do with the software. Like the software is really hard. And so, you know, we had to make it easy and make it cheap, make it scalable. But so that's one thing. Now here's the other one. Let's say we have a microbial sample. Let's say you have a bunch of like bacteria living together, or you know, like a plant that's been infected with a fungus, or some kind of mixture of stuff.
Last week on the show, I brought up a story about a rust, a fungus rust on a plant.
Mm-hmm.
So something like that.
I got pictures of that in my slide deck. But so yeah, that kind of stuff happens all the time. Like things generally don't hang out on their own. Like everything is always touching everything else. So you have a bunch of genomes and they're all mixed together. But if you know which ones are touching, because they were touching in the very beginning of the experiment, you know which ones came out of the same cell. The little touches were trapped before the cells were all broken and the DNA was all mixed together. Right.
So they were part of either the pathogen genome or, say, the plant genome.
Right. And so what you can do is it lets us separate genomes from these complex mixed communities. And what that means is, like, if you go into a soil sample and you scooped it, there's thousands of bacterial species living in there. 999 of them would have never been sequenced before, right? That's the cool thing. So the company's focused on discovery, right? The technology is all about it's all about discovering new stuff and discovering more stuff. Yeah. And you know, whether it's like what's going on with your genome, like a piece of your chromosome flipped around, our technology will tell you that, whereas normal sequencing can't sometimes because it's the same sequence. It just flipped, right? But that might cause cancer. If you're going into a bacterial, a microbial environment where there's a bunch of bacteria nobody's sequenced before, right? Remember, like you have more bacterial cells in you than human cells, right? By a lot.
Yeah.
And and the environment is like a wild, wild west. Nobody knows what's out there. The ocean, like water, like people, like there are these crazy things where like the viruses in the water in the ocean are one of the they play a role in the carbon cycle of the Earth, because those cyanobacteria that suck up the carbon out of the atmosphere, bacteriophages kill them and they sink to the bottom and they become like coral and stuff, right? And so viruses in the water, of which there are uncountable amounts,
untold numbers,
play a role in actual like the global warming, like kind of climate change type carbon cycle. So
that's amazing.
And there's so much out there we don't know. And so the whole point of this technology and why we were so excited about spinning it out is like we just discover stuff by the hundreds. Like every time we process a sample, maybe it's like somebody's poop, maybe it's some soil, maybe it's water, maybe it's coral. Coral is like a little sponge full of bacteria, right? Like whatever it is, there's always new things in there. There's always tons of new bacteria, and you couldn't have found them unless you had some way to separate it all apart.
So wait,
right. It would just be a bunch of DNA otherwise.
Right.
So you can tell where they were touching even after the fact? That's
right.
Because normally if you took like these samples, we're talking about the fish sample, right? We have a bunch of skeletons of different types of fish and things that we already know about that could be in there that we're comparing our sequences to. So we took all these, we have a bunch of books with the sort of in gray, all of the lettering, right? Mm-hmm. We do the sequence and we can fit this here, this can go here, this can go, and we paste them in until we have enough of a recognition. If we don't know what's in there, we can't identify anything novel, anything new, because we haven't tagged it. Even if you know everything going into a sequencer, you tag everything, so when it comes out, you still have identified what
you can get some information because things are similar to each other. So you can be like, I don't know exactly what strain this is, but this is probably a carp or whatever, right?
Okay.
Like, because it's got a bunch of carp genes in it. Like they're not the same as every So what you're
for a carp, but when you get smaller, then when the genome gets smaller and smaller and smaller, then it becomes impossible. It all looks like noise. So this technology can actually separate and distinguish that noise from. How small of a piece are the reads? How long of a read do you need? I guess.
So the reads we use are actually the short Illumina reads.
Wow.
And so we can use long reads too. We work with like all the sequencing technologies, but it works differently in different things. But that's kind of the idea. The idea is like if you know the 3-dimensional distance between things and what's touching, you can do all this stuff with it. And that's really our contribution. Again, like we didn't invent Hi-C, but all this stuff about separating populations and like putting genomes together, like that's our shtick, right?
So from that, considering how bacteria
impressive, by the way,
also have horizontal gene transfer, when you're looking at the fungal species, bacterial species, microbes, viruses that are trading things back and forth, I guess you're still just looking at individuals, right? Because you're looking at the genes, the chromosomes that are in the genes of the individual cells. But can it tell you anything?
Yeah, kind of. It's not completely single cell because like if you have two of the same one, it's hard. Yeah. The sequencing doesn't know like which one it is, but kind of, right. It's sort of the right thing. So antibiotic resistance in bacteria is a huge problem, right? Mm-hmm. They're saying it's going to kill more people by 2050 than cancer. Yes. And part of the reason why it's so explosive and like almost all the drugs don't work anymore is because antibiotic resistance travels on little pieces of DNA that are called mobile elements. They could be plasmids, which are these little circles of DNA. They could be viruses, which they pinch off pieces of DNA and jump around, right? But they're mobile elements. It's horizontal gene transfer, stuff like that. And And yeah, let's say you have two bacteria. And so what ends up happening is one bacterium will, like, let's say, have a bunch of these antibiotic resistance genes floating around. And there's a bunch of other ones that don't. And then you hit the population with an antibiotic and everybody dies, except for like the guy who had all the plasmids, and it starts sharing them. It like they jump around. So through horizontal gene transfer, they can move to different bacteria, and now all of them become resistant as well. And so you have these sweeps. And so understanding mobile elements, this DNA jumping from species to species, is super important. There's no way to do it by sequencing. Like, let's say you sequence a bunch of bacterial soup. How do you know that this virus was in bacteria A or bacteria B? You can't tell.
Yeah.
That's one of the things that our technology can do. And it can tell us how these mobile elements are distributed across the populations, things like that. So there's like all these kind of funky tricks we can do. In fungi, there's a lot of examples of so maybe should I steer the ship back to
Yeah.
We can talk about the fungi February and the fungal work that you're doing.
So one of the things that has been really fun is fungus. Put the fun in fungus.
That's right.
so within this microbial soup, right, there's also like the fungi are a super important and super neglected community because they're just hard to study. There's tons of them out there. Like if you take a scoop of soil and remove the plant material, like 80% or something, I don't know, 50%, 80% of what's left is fungi. Like they're just they're everywhere. They are the largest non-plant source of biomass. Almost all 90% of the plants out there have symbiotic relationships with them. They're called mycorrhizal relationships. The little roots require each other. The largest creature on Earth is a fungus. This is just a fun trivia fact. Like the largest single creature on Earth is it's a honey mushroom that is 2,000 acres big.
Oh, wow.
Remember, the fungus lives underground. Like the thing you see, the mushroom, that's just like the apple from the tree. That's just a little reproductive thing. The actual creature is underground, and it spreads across multiple forests and stuff, and they can sequence them from different forests. They can tell it's the same dude, right? And so they're everywhere. They're super important, but they're hard to study. We know so little about them. and so some of what we do is with fungi. And so they have these crazy biological things. Another fun thing about fungi is that they were one of the they're an organism that humans domesticated very early. That was one of my, when I think about that's a fact that just kind of makes me kind of happy, to think we domesticated fungi. They're like cats and dogs. Like my pet fungus.
The white mushroom.
Yeah,
the criminy mushroom. Right? Oh, okay.
We make bread, we make beer, we do all this stuff, and we've been doing it for tens of thousands of years. And that's a domestication of fungi. And there's all these crazy tricks they do, like the rust. You mentioned rust, right? So there are many rust organisms out there, and what they do is they infect. Fungi are super good at killing plants. Fungi are great at digesting plant cell walls and killing plants. So there's a lot of plant pathogens out there, and these rusts, they do enormous amounts of damage. There's one we wrote a grant about this where one strain of oat crown rust, in 2012 or something, killed half of the oats in Minnesota.
Wow.
They can go crazy. They can do crazy damage.
Yeah, and, but it's also though fungi expressed enzymes are also responsible for your pre-weathered genes. Your pre-stressed genes also are thanks to the fungi enzymes. So yeah.
And about 15% of all vaccines are grown in yeast, which is a fungus.
Yeast is the most amazing worker.
I did 20 years of yeast research. That was
never complains.
My sourdough starter is very excited about this conversation.
That's right. Yeah. Everybody's sourdough starters are very excited about this conversation.
They're like, my ears are burning. What's that? Bread?
Yeah, you can get yeast to express just about any molecular compound that you want to use for anything. They're just amazingly compliant.
Yeah, that's about right. If you guys want, I can show you. So what we did was, we were doing all these fungal projects, and they were super interesting in different ways. And we started like a little contest on Twitter. It's not a contest. It's just an event, a little raffle giveaway. We give away like branded socks. We give away like free products, stuff like that. We called it Fungus February, and the idea was scientists, post pictures of their favorite science fungus, and then somebody like wins a thing. Let's see. Do you want me to share slides? How do I do this?
If you have slides, you may certainly share them. You can hit the share button at the bottom of the screen. There it is.
Oh, fun with fungus.
Yay!
Yeah, I wasn't sure exactly how much you wanted me to talk or not about stuff, but so I live in the Pacific Northwest.
Ooh, pretty.
So like an amazing place to, and we also do a lot of mushrooming. That's a side thing, unrelated to research. But so we did Fungus February at some point, and so we did this little Twitter contest where we're like, do you guys like fungus? You like to do fungus science? Post up pictures of your favorite science fungus, and somebody's going to win something. And people started posting like the most amazing stuff. Like, so this is a rust right here. I'm assuming you guys can see this. I didn't.
Yeah.
This is the like good old Saccharomyces cerevisiae, the brewing yeast that is the workhorse of a lot of research. And some of them are like, some of them have these like multinucleate cells and all sorts of other stuff. Here's some other funky ones. They like, some of them will glow in the dark. Some of them have these crazy multinucleate cells, lots of different genomes in the same cell. Like they'll mix tons of genetic material from different strains, or like they'll endoreplicate themselves a lot. So they make this gorgeous stuff. Some of them will like wear crazy pixelated shades. You can see this one. So yeah, we did all this stuff, and like so much interesting biology was coming out of it. We work not, like as I mentioned, we work not just in fungi. Like I have a little video. We work on all sorts of organisms. We've done genomes of like, I think possibly thousands of things, hundreds of things at least. Plants, animals, mosquitoes, cannabis, like fruity things. This is what the technology is, like I was telling you about. Like you kind of, right, stuff curls up and then you trap the little junctions between the sequences that are touching, and then you can tell what's far, what's close, and you can separate mixed DNA. And so this is a rust story. So one of the cool things about rust and its biology is that they do this thing where different strains will actually mate with each other. And the nuclei, which is like a nucleus is where the DNA is, right? And so the nuclei of the two will just hang out in the same cell. And so you basically get a strain that's like two strains in the same cell.
Okay.
And depending on which combinations of things are in them, it determines like how virulent they're going to be. So this was a publication from a group, well, it's a lot of people. Most of my collaborators are in Australia, but there's this UG99 strain that's like the deadliest wheat stem rust pathogen. And what imagine you're sequencing a strain of fungus, and you need to figure out what sequences are in nucleus A and which ones are in nucleus B, right? Because you got to figure out like what's in the red one, what's in the blue one. But how do you do it if you sequence it all together, right? And it's just very hard because you have they kind of look the same. They're in the same cell. Like you can't tell them apart. And that's what our technology does, is it separates stuff out. So they were able to separate out the two. They're called haplotypes or subgenomes, or there's a couple different names for it. but actually separate the two different ones. And what they found was that half of it, like one of the lineages, let's call it red, was a known one that was like sequenced a long time ago. It's from Africa. And the other one, the blue one, was a new one that they didn't really know like where it came from. So
oh wow,
you can try to understand what genes they have and sort of how they contribute to virulence. This one we just did. This is just a fun one. I don't have like a cool science finding. There's a fungus out there called mummyberry, which it affects blueberries.
That just makes me sad right away. I love blueberries.
But it does it through the craziest mechanism. What it does is it has like a bee or some other pollinator will carry like little fungal spores, and they will land on the flower. Like the flowers have this tube in the middle, right? This stylar canal, and it'll land on top of it. And it'll basically act like a piece of pollen, because normally what happens is a piece of pollen would land over here, and it makes a little root that goes down into the ovary, and then it fertilizes the ovary, and that's how you get a berry. Well, this thing lands up there. This is a fungus now. It's not a pollen, but it makes like a little tentacle that goes down. And it acts like a pollen. It makes a little tentacle that goes down, infects the ovary. And then when the flower turns into a berry, it becomes one of these mummy berries. It becomes like one of these like little possessed, like a little demon possessed.
So it's no longer a blueberry; it is a mummy berry, and when it pops, it's going to throw more fungal spores everywhere.
There's a bunch of spores, and then the cycle repeats. So we assembled that one. It's got all this cool biology. Here's another. I'm kind of going back to the separating things. So yeast, which is everybody's favorite lab organism, well, my favorite. This is what they look like under a microscope. They're super cute. These little things they have, they're called bud scars. So they bud. They like pinch off the cell.
Little tiny baby yeast.
And yeah, they kind of look like babies. I don't know why. Maybe it's like they look like eyes or something. But every time a baby cell falls off, they leave one of these little bud scars behind. You can actually age. You can actually tell how old they are by how many bud scars they have.
Fascinating.
Yeah, but what they like to do is they like to mate with each other. So they have two, like, they're called mating types. You can kind of like genders. They're called A and alpha. And when they come next to each other, they secrete like little pheromones, and then they make this thing called a shmoo. And this is a scientific, official, super scientific, technical term. I swear. They shmoo because they can't move. Like they can't, like not like bacteria that have like little hairs and stuff to move around. These guys, they just reach towards each other, and then they do like they have a little kiss, and then they go, and then they become one.
And then it's
my little shmoo.
The ultimate, yes, it's the most romantic thing you've ever seen.
We will become one.
Yeah, and then they make a diploid, which has two genomes in one. But what's cool is this doesn't have to be the same species. So there are different species that will mate with each other, and they will make these hybrids. In fact, this is super common in your life, because every lager is made by hybrid yeast. In fact, it's like our normal Saccharomyces cerevisiae, the brewer's yeast, mated with this one called Eubionis. This is what they discovered a few years ago. This is what it's like in the wild. Can you imagine? It's growing on the side of a tree in Patagonia.
It looks it looks like little fruits or like oranges or lemons or something that somebody glued to the bark of a tree.
Yep, exactly.
That's fungus.
How did it get to Europe? Like nobody knows how it got to Europe. But this is like the mating of these two things is how you get lagers.
I love a good lager.
So fungi, like they'll mate with each other, two different species. And so we did a little experiment. When we were first starting out, you asked me kind of how we got into this startup thing. And this was one of our earliest projects. When we started working on it, we said, we're going to go to an open fermentation brewery, right? You go to a brewery that makes beer, and some of the microbreweries, they use like natural inoculation. Basically, they leave their stuff hanging out, and it gets infected by something, and then they like, if it tastes like decent beer, they'll bottle it. And so we went to such a brewery called Epic Ales down in South Seattle, and we got some of their open fermentation beer. It was awesome. And then we did science to it. We like, give us some sample out of that barrel.
Beer is science, and then you did science to the beer.
Yep, and
This
we
is
science
just full
that
circle.
beer.
Good.
And so this is like a data version of what I was telling you about separating a mixed community. This is a very simple mixed community. Beer normally doesn't have like hundreds of things in it. It usually has like one or two, but open fermentation beer is a little bit more complex because they have like a bunch of bacteria and stuff too. Mm-hmm. So
when you say open fermentation beers, for the people who don't know what that is, does that mean they're just leaving the lid off when it's fermenting?
Like
so anything.
Yeah.
Yeah. Okay.
Yeah.
Yep.
And so this is like, it's like a craft brewing thing. It gets infected by something out of nature, out of the air, and then you kind of brew it up with that. Well, this one, so what you're seeing, these clusters, is like, it looks kind of like a soccer ball. So the big spots, these groups of colors on the soccer ball, these are DNA sequences that in that sample were all mixed together. But our data, our special, this proximity ligation, this Hi-C data, it is connecting the pieces that were touching each other and separating them into these groups, right? Mm-hmm. So like all of these pink ones, like they were all touching each other a lot, which means they all came out of one organism. And this, same thing with this orange one. And so you separate the genomes out of this mixed soup, and now you can be like, I got this green genome, and I got this brown genome, and I got this. So you can tell which organisms live there, even if they've never been sequenced. And here's one of the cool things. So actually, like four of these bugs, like we gave them names, but they actually were not sequenced before. But especially this green one was an interesting surprise. It was a yeast, but its genome was double the size of a normal yeast. And half of it was mapping to a known organism, and the other half was not mapping to anything. So it was like half, like the genome is double the size of a normal genome, and half of it is known, and half of it is unknown. And so we said, well, maybe this is a hybrid. Maybe this is a new hybrid that nobody's discovered before, and it totally was. So like, because it's beer, it's easy to grow these things, like it's culturable. And so it grew in these like super cute velvety kind of colonies. So for those of you in the audience who don't know what this is this is a petri dish full of like Jello that like yeast will live on. And you put a few of them on there, and one cell will like land here or there. And because they can't move, that one cell is going to start growing and multiplying over and over and over until it builds this little mountain called a colony. But they're all like just like lots of cells that came out of the same clone. So it makes this like cute little colony, and we can look at them under a microscope. They're like really cool. They line up in these like little palisades, it's called. Like they're like side by side growers. I don't know why, but that's not like normal. Like it's
interesting.
And it turned out indeed that this thing had two genomes in it. One genome was from a known organism. One genome was from an unknown organism. And when I talk about this in scientific conferences, I present this data to show like, how else would you be able to tell from sequencing a mixed community that there was a hybrid in there? Like, how would you know that it's not just two different species? Like, how do you know they were inside the same cell? And that's like
you wouldn't know.
Technology does.
Yeah. So away from the, I guess, more, it's still going to be fungally and microbially. There was you made a comment about poop earlier, just on the fly. You have worked with cats, and
I have worked on many poops.
Yes.
In fact, one of our most favorite poops. And if there are any bioinform magicians in the audience, we actually sequenced the poop of a famous celebrity cat named Little Bub. So a few years ago, we had another kind of Twitter thing called #MeowCrobiome. And we wanted to sequence. Yeah, so we got Little Bub's poop, and we sequenced it, and we discovered a bunch of new bacteria. And it's on our website. You can download it. It's for educational purposes. We didn't publish it. It's just for anybody to play with if they want to. So Little Bub, unfortunately, has passed away recently. But yes, but her poop DNA sequence lives on.
Did you find anything interesting? I mean, was there any kind of interesting insight about an internet celebrity cat in particular?
Well, let's just say that what we found, I think, is what a lot of pet owners like. There were a lot of new bacteria in there, but there were also a lot of like known human bacteria in there. And I think somebody was like, can we make the case that like pets and their owners share stuff, which is probably true.
Not necessarily just on the outside either.
Yeah.
Yes.
Correct.
Okay.
Yes.
I have a couple questions. Back to the Shark Tank version of the show where we try to find out.
Okay.
What do you use to sequence with? Are you doing the sequencing, or are you getting the sequence?
Well, we own sequencing equipment of our own, but we also like outsource sequencing to providers who have the like mega machines.
Okay. So, but do you use whatever? Are you using Illumina?
We use Illumina primarily.
Okay. So here's the question: like, is this cost-wise more efficient than going through and tagging everything on a robot before putting it in a sequencer? Like, because it sounds like you don't really need to have anything identified if you're doing a bunch of short, you're taking 400 microbial samples and putting it in. Normally you have to, actually you can do more than that. You have to label each one with this sort of tag that's associated with it that gets read out of the machine later, and it goes through a whole process of algorithm interpretation and looking for that tag and making sure it's associated with the thing that went in the first place. Can you just bypass that whole part of the system?
To some extent, yes, right. So normally what you would do is you would separate the little bacteria in it physically and like grow them up separately and then sequence them separately and say like, But so like, that sample I showed you had 8 bugs, but like if we take a soil sample, it might have 800. So you now have to sequence 8, you have to grow 800 things and sequence
separately though.
Separately.
Because you kind of have to have them separately, tag them, so when they go into the thing, they come out as knowing what it came from in the beginning. You don't need to do any of that.
Right, and keep in mind that I'm glad that I'm blowing your mind.
I love this.
And keep in mind that when you're trying, the problem with the microbiome stuff is that most of the stuff out there won't grow. Either we haven't figured out how to culture it properly, or it's unculturable, the people call it, or it requires other members of the community to survive, and so it will never grow by itself. Right. And then there are practical things like there are these filamentous fungi, like you can't separate them. Like they grow as clumps, these snowflakes and stuff like that. You can't get a cell off. So there's lots of other issues. It's not perfect. Sometimes if a community is simple, if you're just trying to figure out like which strain of E. coli is in your chicken or whatever, like you don't need this stuff. It's heavyweight. It's designed for discovery things. Because we'll go into like a soil sample. We'll discover like hundreds of new bacteria, hundreds of new viruses, a bunch of new antibiotic resistance genes, where they're all it's like more information. Like if you want that information, cool. If you're doing a diagnostic, you may not need that. You just you're trying to be like, is this like a pathogenic thing or not a pathogenic thing? And that may be enough, right? So, but for discovery efforts, like that's kind of what this is geared to be.
Yeah, that's fascinating. I'm very impressed with the applications or the potential applications of what you've got there.
Yeah,
well, this would be a great thing to do. Yeah, with the Martian cores. This is immediately what I'm thinking about here, because we're always talking about how no matter how clean the stuff is that we're sending up there, there's always stuff that we're sending up there. So this would be the perfect way to do that, right? Is you could sequence this Martian dirt, and you'd be able to pretty quickly parse out if there's anything that you haven't seen before versus things that you have.
Yeah, I mean, assuming something like is growing
there. Yeah. Something's actually there.
Because we always accidentally throw.
Well, we do find something. So we discover there's like Carl.
Cyanobacteria. Carl. Let it sit for a while and then sequence it again after a couple of years and see what it did.
Yeah.
I think you'll find Carl, who works in the clean room back at NASA.
Exactly.
And you'll also find, oddly, Carl's cat. was also represented.
Yeah,
Yes.
that is another thing. Yeah, is that like everything is contaminated with human
As
DNA?
well,
Yeah.
because at some point, everything will get there. Yeah.
Just be like, this bacteria came from Matt Damon's poop.
Yes. Perfect. Yes.
What's it doing? Is this an abandoned potato farm? Like,
Yeah. So you've also worked in synthetic biology, and there's this idea of like one day we're going to be able to put DNA printers on Mars and print our own special bacteria that will live there. But, and if you're thinking into the future of where your technology, the stuff that your company's doing, and like the future of this chromosomal genomics, like where do you hope that it's going to go? The kinds of discoveries or things that it might lead to?
yeah. Well, I mean, there are many. I mean, the general trend is that we are moving further and further into like the information space. Like not to get too kind of like trippy, but like life is basically information, right? Like it's
just math. It's just life. The whole universe is just math.
Basic. Math is applied information.
We emerged from math. That's
and so the more we understand how everything works, everything becomes better, right? So like the most applied things are like new diagnostics, new medicines, trying to understand what the mutations are in your body and how are you sensitive or resistant to particular drugs, customized therapies, customized medicines, that kind of stuff. In the agriculture space, like what we were saying, like plants need fungi, plants need bacteria. Nobody knows what's going on in the soil, but they know that like there's like good soil and bad soil. Like sometimes farms will fail. I'm trying to understand sort of being able to, let's say, predict what crop is going to grow best over here. What crop is going to grow best over there? Trying to be like, we found like a really good combination of microbes that we can spray on the crops instead of pesticides, and and use and they do
that would be cool.
There's a whole
thing that people are. It's a big area of development in the agriculture space, using natural bacteria as fertilizers and pesticides instead of like chemicals, right? Nitrogen fixation.
Awesome.
Bacteria, there's tons of nitrogen in the air, right? Air is like 70% nitrogen, but like plants can't consume it. They need bacteria to turn it into like ammonia, basically, to fix it, and then plants can use it. That's why we spray ammonia like fertilizers on plants all the time. But what if we didn't have to do that? What if we just got a bunch of bacteria that did that more efficiently, and now you have better plants grow better without having to even necessarily do like GMO stuff, right? So
probiotics for plants. I love it.
Yeah, I mean, like I'm not like when I think synthetic biology too. Like synthetic biology usually refers to using like bacteria or microbes to make stuff. So like using them to make things. You guys know like Impossible Foods, right? It's the company that like they make meatless burgers, right? Well, what they've learned is that the trick to making a meatless burger taste like meat is making it taste like blood. Yeah. And so the trick is to make it, and so they synthesize their hemoglobin. They make heme. They make like a version of heme
out of fungi
in yeast. Yeah. And so yeast makes the heme that then goes into these plant-based burgers, right? I mean, there's like so much stuff. Like some of it is like super crazy. Some of it is super practical. But I think the whole thing is pushing us from like a lower information state to a higher information state. We like know more about everything. We can do all sorts of cool things.
I think that's fantastic. I want to know more about more things. Take us there. Take us there, Ivan. What is one thing, if you want people to have a takeaway message about genomics and about the technology you use, what's like the one takeaway message you hope people will take with them?
I think that people shouldn't be scared of it. I think it's very easy to make science scary and confuse people with it. Everything from like vaccines to GMOs to like all this other stuff, and I think people should remember that like making harmful genetic things is harder than they think. Like you're not just going to magically release like super stuff. and the information that we can get and the benefits of getting the genomic information out of systems is super valuable. And so the message is there's an enormous amount of stuff out there. We've barely scratched the surface of what we know, and we should like keep trying to learn more all the time.
That's something I'm seeing here that I feel like was brought to light for me from this interview was every time I see an article pop up that, oh, we sequenced another genome, it's like, okay, cool, I guess, nice.
Yeah.
But now it's much more clear to me how important that is, and how knowing things. It's like the conversation we're having about the null hypothesis. Yeah. Knowing what you know allows you to know what you don't know. If that makes any sense at all.
No, it doesn't.
It does.
So, I didn't get it. But,
it does. It gives you more ideas of things. What else? Oh, it gives you more questions.
It's true.
This is a thing we know. This is
real
a thing
concern
we don't know.
In being like, okay, like we're sequencing this genome, why? And it kind of like goes back to what you guys asked before, like how do you do it? So sometimes people do these genome projects just to do them, and then figure out, because there's a little bit of a chicken and egg. Until you do it, you like don't know what the question really is, and so sometimes, but so
the problem I always have is that doesn't that end up with too much information? Like if you didn't like, I'm going to zero out everything but this narrow field. There's so much data that how can you distinguish noise from not noise? It's just too much, right?
It is too much, yeah. And you have to figure out some way to take important insights out of an overwhelming amount of data. I mean, I think probably like, I don't know what it is, like every month we generate like 10 times all the books that have ever been written,
Yeah.
Like,
it's a ridiculously large amount of information to be able to sort through.
And then what do you learn from it, right? Like, because the problem is, and this is a problem for our field, for the genomics field, we can generate sequence data way faster than we can figure out what it means. So, because to actually figure out like what does this gene do, you got to do like experiments in the lab with cells and maybe mice and maybe people, and maybe you have to like test stuff, and that takes forever. Sequencing and doing computer stuff is fast, right? And that's sort of the race.
So you can generate all these things, but until like these genes have been annotated, until somebody can put a name on it, and not just a name, like we'll call this gene Fred. Okay, Fred's a gene now. What does Fred do? Nobody knows. We know Fred's a gene, but now you have to go to through experimentation and knock out for one specific gene to know what its effect even was on the larger body.
Well, keep in mind that gene Fred may not do anything. It may only do things in conjunction with 5 other genes, right? That's the problem. And it's like how much of gene is being expressed? Does it need to be a lot or a little? Like cancer is all about good genes going bad. Like there's no like gene that was designed to make cancer, right? They all supposed to be good things, and then you like change you change the little function, and now it's got a completely different action, right? So
and it's only bad for the larger organism. It's not necessarily bad in the terms of the minor ecosystem of the tumor and all that kind of stuff. So it's all perspective.
Yeah. Now it's existential stuff where we're like, does function even mean anything? Like,
what does it mean? Yeah.
Can it position it on a chromosome?
Sorry?
Can it locate the position on a chromosome, or what?
Can what locate?
What you're doing as well, like tell you where it is on the chromosome, next to what, and where all the answers. This is okay.
Yeah. So suddenly you start getting those. You start getting genes that work in conjunction that are local to each other, or you can start if you start. Started doing association studies.
Level. It's not even DNA anymore. Now you're talking RNA, you're talking proteins, you're talking metabolites, all this other stuff. So I mean, I'm just talking about like the blueprint. Like I'm just talking about like where stuff is. Like
the raw ingredients
is like a whole separate dimension. And
Ivan, thank you so much for joining us tonight.
Yes.
Yeah, thank you so much for having me, guys. It was great.
Where can people find you if they are curious about Phase Genomics? You can find me
at phasegenomics.com. You can find me at my name, #@Ivan Liachko on Twitter. if you are interested in genomics and startups, we also run a little event sometimes called Genome Startup Day. So that's just genomestartupday.com. We bring a bunch of genomics startup founders. We talk to them about their experiences, things like that. And so, yeah, that's our website. If you're interested, we have a whole section on publications and how it works. And we have like a news articles and blog. There's a lot of stuff on there. You can follow us on Twitter, LinkedIn, the usual social media type thing, except for not Facebook. I don't think we do anything on Facebook.
It's probably better that way.
Don't make the Facebook overlords mad. You never know.
I'm not saying anything. I'm not saying.
Kiki. Quiet, Kiki. Thank you so much. This has been absolutely wonderful. Yeah, really great getting to speak with you.
And thanks to my good friends at S2S PR who connected us. Eric.
Yes. Many thanks to Eric for connecting us for this conversation. It was, it's been really, really fun getting to find out about your technology, getting to find out about how we can delve into
business.
Genomes a little bit more. Let's get to business. All right, we must move on with the show. Thank you.
See you guys. Thanks again.
Thanks.
Have a great night. Bye. This is This Week in Science. We are talking about science. We have a bit more of the show coming up. We have stories ahead. For those of you that are enjoying the show, hey, make sure you click that like button, subscribe, get notifications for when we go live. And if you would like to help TWIS grow, please get a friend to subscribe today. If you're subscribed, maybe get a friend to subscribe too, and then you can share the show together. It's amazing. You talk about it together. It'll be fantastic. More the merrier. Thank you. I think there's a thing happening right now. Oh, wait, I'm getting a broadcast. It's coming in loud and clear because I do believe it's time for Blair's Animal Corner with Blair.
Loves are creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
With Blair.
Thanks. So disarming. Anyway. Hey, it was Valentine's Day last weekend. Roses are red, violets are blue. You know the saying, if you chew off my wings, I'll chew off yours too. It's tale as old as time.
Okay.
This is a study about wood-eating cockroach couples. They are monogamous, these cockroaches. They live in Japan. This is a piece of research from Kyushu University in Japan. And a couple of researchers, Haruka Osaki and Aiti Kasuya, they happened across some wood cockroaches that had chewed off wings out in the wild. They were very confused by that. And so they decided to bring some into the lab to study. Now, wood-eating cockroaches normally feast on fallen trees in the wild, and in more recent days have, of course, taken to eating wood that makes up people's homes. And while they were studying some of these bugs in the wild, they noticed that most of the adult roaches had wings that were almost entirely chewed off. So they took 24 young adult pairs with wings still intact and brought them into the lab to study. It appears as though these cockroaches use wing chewing as part of an after-copulation grooming practice.
I love you. I will chew your wings off.
That was really great. I had fun. Did you have fun? Great. Let's chew each other's wings off. Usually, one of the pair was groomed in such a way after a mating session, but it varied how much the chewing would happen between one and the other. In some instances, one would eat the wings and then the other would kind of stop, and then just leave. In other instances, the one being chewed on would shake their body to let the other one know, okay, enough. That's enough. You took enough of my wings off. Thank you very much. Out of the 24 pairs, 12 chewed each other's wings down to the extent that they had seen in the wild. So this seemed consistent. As I mentioned, they are monogamous. So the prevailing theory now is that this is actually a form of grooming, that cockroach wings are susceptible to mites or mold. And so eating wing tips could be a way to remove mites or mold to ensure survival to a later date.
But now you can't fly, and
right.
you don't have wings. But hey, no more mites. You're clean now. It's grooming gone too far. It would be as if it were, monkeys grooming each other, but instead of just grooming off the ticks and mites and stuff, they're actually pulling all the hair off.
I mean,
people
I'm not going to yuck their yum here today. Say, you know what, cockroaches, if that's what you do,
you do you, cockroaches. You're such a good person, Blair.
I'm not going to king shame cockroaches today, is all I have to say about that. But
Great.
Happy Valentine's Day.
Thank you
To those cockroaches, especially.
I love it.
Now moving on, I have a much more complicated story about spider legs. So, a lot of people are afraid of spiders. There's a lot of theories where that comes from. There was a really cool story we did years ago now, I think, where it seems like there's actually a genetic inherent fear of spiders in humans from a very young age. But a lot of people will cite the way spiders' legs move as the problem that creeps them out. They move around almost as if they have a mind of their own. What if they do, though? So a recent study looking at Araneus diadematus, which is the common garden spider, otherwise known as Charlotte from Charlotte's Web, looked at
I love Charlotte.
How they make their webs and what part of their body is really in charge of that web building, which is a really complicated process, actually. So in order to figure that out, researchers actually, over many, many years and many, many experiments with spiders, they were able to make a virtual spider. There's no other way to say it.
So this is a spider in a computer simulation. Right. It's a simulated spider.
Yes, Theseus is the spider's name.
Great. Okay.
They ran Theseus through countless numbers of webs that they scanned. They took pictures of from the real world. So they were picking real spider webs and then kind of teaching Theseus the shape of these webs. So then they were able to set Theseus on the kind of the web scaffolds that a spider would make. So spider webs are made out of a bunch of different types of webbing. And so you have kind of the main scaffolding pieces of the web, and then they make that final last pass of the sticky sensory webs on top, which is the real trap, the real way that they catch their food. And so they were able to send Theseus on this final route of building the web, all based on learning from the real spider construction. And what it looks like is actually an iterative process where they don't just go around in a spiral pattern. If you watch a spider build a web, there's a lot of turning back and forth and zigzagging, and it almost looks nonsensical, but it's actually really specific. It's in order for them to fill out each of these individual holes, because a spider web, if you think about it, is not a perfect circle.
No.
So that means that their scaffolding pieces are not always symmetrical. So if you have this kind of elongated web, then some spaces in the elongated portions are going to be bigger than spaces on the sides that are smaller. And so doing some zigzagging in those larger bits will make sure that they have covered the entire surface area of the web with evenly spaced out sticky sensory webbing. So that means they have to make these these decisions in the moment. These iterative decisions. Okay, I went here. Now I have to go here. Okay, here, this needs more. So now I need to turn back and forth here. All right, so now I'm going to go back over there and I need to fill this part in. So it builds upon itself.
Too big of a gap here. I need to fill this up. This isn't the way I want it. Oh, this is looking pretty good. Maybe I'll keep going. Nope, got to turn around.
Yes. Yeah. And so what they found was that this is actually something that the brain has outsourced into the spider's legs. The spider legs build the webs semi-autonomously.
Okay.
They are closed feedback loops inside of each leg.
So the legs are basically feeling
tension detectors. Right?
Yeah, they're feeling the distance, they're feeling the tension, they're plucking their violin string, going
stability,
ding, 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 ding.
resonance, something's not right here, it needs another pass.
But it's not the spider deciding, it's the spider's legs deciding, sensing and reacting. Wow.
Yes. And so they also confirmed this by figuring out what happens when spiders lose legs. So when a spider leg becomes trapped, they'll ditch it. And then they regenerate when they molt and they get their next exoskeleton.
Wait, so what? I didn't know that
spiders...
Well, how did I not know spiders do that? They grow new legs?
Crabs do it too. Stuff with exoskeletons do this because they don't have any internal anatomy that stops them from doing it. So basically, they're just rebuilding an exoskeleton. So you can do that. Yeah. That's easy.
And especially if you're growing and what you do is you're rebuilding an exoskeleton by molting all the time. It's what they do.
Imagine if every few months you built a whole new set of bones. It would be easy to regrow an arm.
Yeah. No problem. What?
So anyway, so when they discard their trapped leg or it breaks off or whatever happens, they do get another leg, but it's usually a little shorter. It doesn't match the rest of the body perfectly. But it has brand new hairs and sensors. And it still is able to build these perfect webs, and these individual legs act in the same way.
So if you go around collecting spiders' legs, you too can build webs. Wait, no, that's not the way it works.
Probably not.
So wait, what does a spider's web look like if they lose a leg and then try to make a web?
It probably looks the same. I mean, they have 8.
I got plenty.
Yeah. But
I wonder.
I think this was a really interesting story. After last week, we were talking about dragonfly brains and how I was talking about how they're kind of rudimentary. So this is part of that, right? Is that a spider brain, this is part of where this study came from. A spider brain, as far as you can tell, is not capable of making something as complex as a spider web. So this helps answer that question.
Because it's not the brain, it's just the legs.
It's they've outsourced it.
Yeah, but there are other ganglia around but that gets at it's like very almost octopus like, where octopus legs have their own brains.
I was thinking exactly that. Yes. It's very octopus like. And of course, the researchers looking at this, what's their next choice after this? Better bring it to robots. So a decentralized system would be perfect for robots. They call this morphological computing, roboticists, and they've only recently discovered its power through looking at the animal kingdom. And now they want to kind of push that towards robots because it could totally change the game if you can take external stimuli and not have to feed it back to a centralized computer.
Yeah, well, I mean, I think for so long, we have had this goal of figuring out the human brain, or, the brain. Humans have this big brain, and so it's all about the brain. But one of the earliest things you learn about in physiology is the reflex arc and how spinal reflexes allow us to walk. We have this same kind of, we're not building webs, but we're able to walk, we're able to run, we're able to have these patterned movements it's basically once you start moving, you have offloaded that cognitive effort to your spine and to the rest of your body.
I must not. No wonder I was so bad at track. The whole time I'm like left, right, left, right, left, left. Oh no, I'm falling.
You're thinking too much. Thinking too much. Justin, oh no.
Oh yeah, so they also say another example of a decentralized task in the human system is your heartbeat.
And your heart, so your heartbeat, you have the pacemaker cells that keep it all on rhythm. And you have your vagal currents that come in and kind of go, oh, slow down now or speed up. You know, you have your other stimulating nerves that come in and make it go faster. But just to keep your heartbeat going, it's got its own rhythm. It doesn't need anybody telling what it to do.
And it speeds up and slows down. It's got it all figured out. But so this is a very cool example of, you know, we thought for a long time, we thought the human eye was the best that eyes could get. Yeah. It's like, oh, wait, actually birds have really good eyesight. Oh, wait, there's mantis shrimp. Oh, wait, nautilus have pinhole eyes that are actually pretty good and are nothing like ours. Yeah. So this is the human brain, that's the best it's going to get. Oh, wait, there's octopus brains that are donut shaped and they have the brains in their arms. And then also there's spider brains that have the...
There's also cell size of brain tissue or neurons or something like that. Right. So that even if you have a giant brain, if you've got giant cells doing that work, versus I think it's birds have very small cells. So they can actually have in that tiny little brain, they can actually pack in quite a
more
few
stuff.
Operating. Yeah. So
and the distances between the cells, it's shorter, and so things happen faster. Yeah, there's a lot of trade-offs.
Levels to the whole what's a brain.
Yeah. We're not the top of this evolutionary pyramid.
Well,
it's a plateau, right? There's all this sorts of stuff that got to where we're at. So I will say that I do think it's always amazing whenever I hear about another bird using a tool, innovative thing. I love those stories. I think especially COVID's super intelligent, much more so than we thought. But I've never had a power drill made by a crow. I mean, I think there's.
Hey,
we're dropping stuff on Mars. I do think there's something pretty remarkable about the human brain that's not quite plateaued.
Sure. Cetaceans are hanging out in the water looking at us on shore going, oh, those dumb monkeys. Thinking they're going to do things again. They're just ruining things.
And that just shows their lack of anthropological knowledge, calling us monkeys and not apes.
Well, Well, we're going to keep this show going. Blair, thank you for an amazing Animal Corner.
Yeah, you got it.
This is fantastic. Everyone, this is This Week in Science. Thank you. Thank you so much for joining us this week. And I hope that you join us every week because we bring you science all the time. And the only way that we're able to do that is with your support. So if you have the ability, please head over to twis.org and click on our Patreon link to choose your level of support today. You can help us bring science, curiosity, or sometimes random facts or arguments about how random facts may have actually come to be or what they actually tell us. But you can help us share that and keep doing what we're doing every single week by becoming a Patreon supporter. $10 and up a month, just a month. We will read your name at the end of the show. Help us grow that list. Help me just feel like I have so many names to read. Like, oh, I want to do that. Make it longer. Make it exciting.
Voice.
Micro machines. That's right. I'm going there. Thank you for your support of TWIS. We really cannot do this without you. All right, Justin, tell me a story. You got a story for us? Or two?
I've got two stories. Let's see. I don't know which one is. Oh, yeah, here it is. From the Okinawa Institute of Science and Technology. They looked at some genes that can help against COVID. So some people are experiencing mild or even no symptoms whatsoever when they are confronted with COVID-19. Others are not so lucky. This is the researchers at the Okinawa Institute of Science and Technology, Japan, teamed up with Max Planck Institute for Evolutionary Biology in Germany, and they found a group of genes that reduce the risk of a person becoming seriously ill with COVID-19 by around 22%. And those genes are inherited from Neanderthals. This is Svante Pabo. Of course, other factors such as advanced age, underlying conditions such as diabetes have significant impact on how ill and infected an individual may become. But genetic factors also play an important role, and some of these have been contributed to present-day people by Neanderthals. So last year, I think we talked about, was it last? Oh, yeah, everything's last year. Last year we talked about how Pabo and Professor Hugo Zieberg, they reported in Nature that there was a greater genetic risk of developing severe COVID-19 if you had inherited Neanderthal genes. There was some set of genes there. So the latest research builds on a new study published in December from Genetics of Mortality and Critical Care Consortium in the UK, which collected genome samples from 2,244 people who did develop the severe COVID-19. The UK study pinpointed additional genetic regions on 4 chromosomes that impact how individuals respond to the virus. Now, a study published in PNAS, Professor Pabo and Professor Zieberg show that one of the newly identified regions carries a variant that is almost identical to those found in 3 Neanderthals. There's a 50,000-year-old Neanderthal from Croatia and 2 Neanderthals from southern Siberia, one that's 70,000 years old, one that's 120,000 years old. So these are genes that are going way back. This second genetic factor has the opposite effect of that first one that they had discovered, providing protection rather than increasing the risk to develop the severe version of COVID-19 reaction. The variant was located on chromosome 12, reduces the risk that individual will require intensive care after infection by about 22%. So this is Pabo again. It's quite amazing that despite Neanderthals becoming extinct around 40,000 years ago, their immune system still influences us both positive and negative ways today. So to try to understand how these variants affected, they took a closer look at the genes located in these regions. They found that 3 genes in this region called OAS code for enzymes that are produced upon viral infection and in turn activate other enzymes that then degrade the viral genomes in the infected cells. This is absolute immune system for this. What I kind of found also interesting is that these Neanderthal would have been sort of just coming out of an ice age in one of the cases. Yeah. So this is like as people sort of reemerged back into the world after a long point of isolation, or these Neanderthals reemerged out into the world after probably some decent isolation, they probably encountered a lot of these viruses. And so there's probably a culling of who had the right gene, who didn't have the right defense gene. They found that the variant increased in frequency after the last ice age and then increased in frequency again during the past 1,000 years as a result. Today it occurs in about half of people living outside of Africa have this gene. And around 30% of people in Japan, which is sort of interesting because last time they looked at the gene that looked harmful, nobody in Japan had it.
Huh. Interesting.
So it also starts to focus you back to, okay, so now you know that you can also sort of isolate which groups of Neanderthals intermixed with which groups of humans at some point in their previous history based on who does or does not have the vulnerability gene versus who does and does not have the gene that procures some sort of resistance or some sort of fight back.
Yeah. I wonder how it also, if it also has anything to do with how COVID-19 is spreading around. So it's only seems to be affecting the severe COVID-19 effects, but not necessarily transmission that they can tell, right?
Well, I mean, yeah. So this has nothing to do with transmissibility. I don't think that was
just looking at symptoms.
Yeah, it's about whether your body takes on an immune response that prevents the cascade of events that take place to get one severely ill where they're personally in danger of dying or increased danger of dying from the disease. But yeah, that's the whole problem with, of course, with something is it would almost have been better if this was just a very, 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 very lethal. It wouldn't be much better if this was a very lethal virus that killed 50% of people on contact.
Nah.
It wouldn't have spread.
It wouldn't have spread as much, maybe.
No, we wouldn't have killed as many people.
You know what? I don't want to think about whether or not it would have been better or worse.
That's where MERS got it wrong. MERS pretty much killed everybody, and so there was no time to spread it. Here's the virus.
People were afraid of it, so they were like, "I'm going to stay home," and they stopped the spread themselves behaviorally because it was much more serious. Oh yeah. I don't want to talk about that psychological effect, but
okay. Well, then let's talk about something else.
Yeah.
So anyway, if you have that rare variant that came from a Neanderthal, it might be making you 22% more immune to the serious effects.
I have like 2.5% Neanderthal genes, so
But are they the right ones? Are they the right ones?
I kind of hope so.
Got a 50-50 shot according to the study. Anyway. If you look it up, though, if you did one of those genetic testing things where you send in your spit and they give you your genome, it might tell you if you have the rare variant. Next study, commercial genetic testing companies are using extremely unreliable technology in detecting very rare variants, meaning results suggesting individuals carry a rare genetic variant, be it disease-causing or not, are usually wrong. And if you're usually wrong, that means you're wrong most of the time. So why this is important, though, is that people have actually gotten surgery. And it doesn't go into it here, but I'm assuming that this is people who had the very rare genetic variations for the BRCA1 that shows increased risk of breast cancer may have gotten mastectomies when they perhaps didn't have the variant.
Oh, no.
So it's created preemptive decisions about health that are mostly unreliable. This is not a good thing. Team of University of Exeter conducted a large-scale analysis of technology that is being used, technology using data from nearly 50,000 people, so that's a pretty decent set. They found that the technology wrongly identified the presence of very rare genetic variants in the majority of cases. So the team analyzed SNP chips. So SNP is single nucleotide polymorphisms. So this is when you have a genome and you've got all the letters, and then one of the letters is switched to something else. Just one. That's a single nucleotide switch. So so they've tested this on specific locations across the genome, and they found that these chips that can test for those single changes that have been apparently utilized when somebody's submitting their DNA to one of these commercial sites was really good. It was excellent at finding common genetic variations. So something like risk of diabetes, type 2 diabetes, these sorts of things. Maybe it's something that has to do with the earlobe or the color of your hair or something like this. They were excellent at this. But when there was something that was a rare SNP change, or rare to happen in nature, the machines were really bad at detecting it, and in fact made mistakes. So like the previous conversation we were getting into was talking about all of the different types of short read, long read, how many layers of a read you get, how you can make those decisions based on algorithms that you apply to this raw data, and putting these pages of these books together. A lot of the commercial technology is using sounds like a rather roughshod version of that. Now, they've also, a lot of them, been around longer than this next generation technology has existed.
Yeah.
So.
But this sounds like this just means we need better genetic testing in medical use. because you could get your 23andMe, right? And you could see, oh no, it looks like I'm predisposed for XYZ. Yeah. If I could then go to my doctor and say, hey, I got this scary thing on my 23andMe.
Verify.
Can you do a genetic test for this disease? I think that's like, you know, your Fitbit might tell you that your heartbeat's bad, but you're not going to do anything about it until you go talk to a doctor first, right? And they're going to hook you up and put you on the treadmill and figure it out with their better tools. We need to have the better, more reliable tools for medical decisions, it seems like.
Yeah. It is interesting, though. I think what might be happening here is that you've got it's a rare variant. And so what you're dealing with is something that occurs in a very low proportion of cases. And so a lot of this genetic, it's on the, as you said, Justin, the bioinformagicians.
Infomagicians.
Yes.
Bioinformagicians.
On that side of things, it is statistics and it's probability of things popping up. And so if you are layering things together and putting them in a certain order and figuring out whether or not they exist in a certain place, in a certain form, all the time or only sometimes, that's going to depend on how many times you're reading something and how many overall copies of it you have. And so if it's rare, the probability of either it being an error and something that was a mistake in the reading or a real signal, that is a real issue. So it's coming down, it's got to be, because it's rare, this is a statistics and probability issue.
Yeah, so they said in very rare variants, these are variants that are present in less than 1 out of 100,000 individuals. They have found that those causing rare genetic disease ones, 1 in 100,000 people would have this. 84% were false positives in the UK Biobank. Mm-hmm. So they had, in the data from commercial customers, 20 of 21 individuals analyzed had at least one false positive. At least one false positive for a rare disease-causing variant that had been incorrectly genotyped. Now, one of the things that they also point out in this study is that a lot of the companies that are doing this commercial genome stuff are also, when they're going back over, and they are taking second, third passes at the rare genetic variants when they show up and correcting them in sort of the final output. Mm-hmm. However, the big problem is if anybody is pulling their raw data or taking the initial information from the read, they will get those false hits.
Yeah, I mean, I think a lot of what this there are a couple of people saying this already in the chat room, which is don't use any of these personalized genetics sequencing companies where you can have your genome read for medical decisions, ever. Verify, go to your doctor, like you said, Blair. Make sure that you're getting a couple of tests. Make sure that you are not just basing it on one data set. Make sure you have verification. Yeah, especially, and there were like lawsuits for 23andMe, like at the federal level, about whether or not 23andMe and other companies could even be used to give quote unquote medical advice. And now they have a whole thing where it's like, this is not medical advice. You should not be using this to make medical decisions. And they say they have disclaimers about that entirely, but
it was shut down completely, and there's lots of disclaimers because it became a very big legal thing. However, that said, for the common ailments that ail people, it's actually shown to be quite accurate. It's
like rare stuff.
The rare stuff is what's getting the false hits. So if the rarer the disease you find when you put in the diagnosis, when you self-diagnose on one of those web medical diagnoses tools, and you go on there and you say you feel nauseous and have some fatigue and a little bit of a joint ache, I can put that in it. And next thing you know, you've got the yellow fever.
It's every time you have cancer.
Or you've got some rare tropical disease. Now, because cancer's pretty common. Got some rare tropical disease. Like, yeah, it's a stretch.
Yeah. I'm going to jump forward. I've got a couple of stories before the end of the show here. I want to talk about your brain.
My brain? Your brain.
Your brain, my brain, everybody's brains. Yes, I want to have a conversation about using your brain. So this is a really interesting study out of the Max Planck Institute for Experimental Medicine and also the University Hospitals of Copenhagen and Hamburg-Eppendorf. All these researchers collaborated on a study looking at the effects of mental and physical demands on the brain. So they discovered a while ago that hypoxia in the brain releases a compound called erythropoietin, EPO, and this is a growth factor. So shortage of oxygen releases a growth factor. And in these experiments that they did with mice in the lab, running in a mouse running wheel or doing mazes and having to do cognitively demanding tasks for a mouse, they discovered that oxygen deficits are created, little localized shortages of oxygen around your neurons that are working really hard. If they're working really hard, they're using up all the oxygen and the blood isn't necessarily keeping up with delivering it. And that's a slight hypoxia. They call it functional hypoxia. And they discovered it leads to the formation of new nerve cells. They wanted to know how, and they discovered then that the hypoxia activates this growth factor, erythropoietin, in the brain, and it leads to the development of new connections, and dendrites is a part of stimulating the brain. And now the big question is this the thing that is involved in making the connection between exercise and mood? Exercise and the stimulation and the effects that exercise has on the brain. Is this what's happening in the human brain? Is it functional hypoxia that creates...
Running makes you smarter?
Running makes your brain expand.
Wow.
This is also kind of, I think also last week we talked very briefly about how stress can be good for a system. So this could be that too. Just any sort of stress, any sort of... kind of fight or flight moment, any sort of challenging moment where you have to think fast, it could create this stress that causes it. It's like growth from hardship, right?
Yep. Yeah, exactly. And I imagine that it's the opposite. So this is an acute problem, right? So if you're running, you're not chronic. Exactly. You're not always running. You're running a little bit, and that's good. Or it's you're thinking really hard, you're working really hard, but this isn't chronic stress, right? It's working your brain a little bit, but not too much. And so this, I think, it's a really interesting mechanism, and their next steps are going to be trying to discover whether or not they can put a person on a treadmill or an exercise bike and actually see if they can measure some of these changes and see if they can, they're able to find in mice, because they can slice the brains and check for gene expression and all sorts of stuff. They're able to find a change in expression of this EPO gene. But they have to try and make this connection in humans as well. But this could be one of those really important connections between how physical activity leads to changes in the brain.
Or as JG in the chat room so eloquently points out, and I like the way you think. So exercise or just hold my breath.
That's right. Just every once in a while, not too long. Hold your breath while you're studying. I don't know. Yes. Breathe, everybody. Breathe. My final story for the night is about birds, because I love birds. And I've always wondered this about birds. How do little tiny birds stay warm? right? How do these little
They have down feathers.
They have down feathers, right? But how do they really stay warm? Like, there's a difference between can you take a bird from the beach and put it up on the mountain, and will it stay just as warm? Will a bird that you take from the mountain and you put it down on the beach, is that going to get too hot? Like, is there a difference in what's happening there? And so Peter Buck Fellow at the Smithsonian's National Museum of Natural History took a look at the Smithsonian's collection of 625,000 specimens to study the feathers of Himalayan songbirds, 249 species. These are birds living at really high elevations. And yeah, it was based all on her question. She was studying birds in the Himalayas, and she said, do they have a down jacket? I have a down jacket. Do they stay warm? So she looked at all these birds that were in the Smithsonian's collection and discovered that, yes, birds who live at higher elevations have more fluffy down than birds who live at lower elevations. So the elevation that the songbird lives at affects the thickness of their down jacket.
It's just like dog breeds from colder climates have thicker undercoats.
Yeah. And so this does kind of get at that question of where different animals can go and where they're going to be comfortable.
Mm-hmm.
And so as we have climate change kind of moving species different places, this may be an indicator of which places birds are going to be more comfortable by being able to predict how they might be able to withstand or respond to climate change very easily. Although this is very likely something that is a very adaptive trait. And so this study does not get at the question of causation. Is this, really a genetic issue that birds at higher elevations have, like people who live at higher elevations, do they have different genes that lead to more of the down? They don't know that.
Probably both.
Or is it just adaptation? I'm cold. I need more feathers.
I would guess it's both.
Yeah.
Because birds are constantly losing feathers and molting and growing new feathers, and unless you're a penguin, then you do it all at once. But
it's all at once, yeah.
That's called a catastrophic molt. But fun fact.
That's a great name. Oh my God, we've had so many fun facts on the show tonight. Catastrophic molt.
But yeah, I bet it's both. I bet they start out with a set of thicker feathers, but that if they were hot, I bet they would lose some density. That would make sense to me.
Yeah. Yeah. So anyway, down jackets on birds. Every time I look at a little bird in the cold, cold weather outside in the winter, I'm going to be, nice jacket, bird. Looking cool, and we're looking warm in your little avian insulation jacket.
The underside is the warmth. The outer side is for looking. It's pretty.
Yes.
Or hiding. Or both.
Maybe both. Yeah. Anyone have anything else? No. We have had a wonderful show. I think we've done it. We have made it to the end of our show. Thank you so much for being here with us. I would love to shout out to friends and helpers of the show. Fada, thank you for your help on social media and manning the show notes and helping out on the YouTube channel. It's just amazing. Gord, thank you for manning the chat room. And I've seen you on Twitch. You're heading over to the Twitch channel recently, heading over there. Identity 4, thank you for recording the show every week. Do appreciate that. Keeps it going for sure. And Jessica, thank you so much for joining us once again. I'd also like to thank our Patreon sponsors for their generous support of This Week in Science. It takes me a while to share my screen again. Bum, 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 bum. Thank you, TWIS patrons. Woody MS, Andre Bassett, Chris Wozniak, Dave Budd, Vegard Chefstad, Hal Schneider, Donathan Stiles, a.k.a. Don Stylo, John Shioli, Guillaume, John Lee, Ellie Coffin, Gaurav Sharma, Shoe Brew, Darwin Hannon, Donald D. Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Bryan, Carrington, Matt Bass, Joshua Fury, Shauna Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, aka Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Melizond, Johnny Gridley, Kevin Railsback, Flying Out, Richard Porter, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood, two fabulous thespians. Love that. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Matt Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavis, Sue Doster, Jason Olds, David Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Gary S., Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support of TWIS on Patreon. And if you are interested in supporting us on Patreon, you can head over to twis.org and click on the Patreon link. On next week's show,
we will be back with on Wednesday, 8 p.m. Pacific time, or Thursday, 5 a.m.-ish Central European time. Broadcasting live from our YouTube and Facebook channels and from twis.org/live.
Hey, do you want to listen to us as a podcast?
Sure.
Just search for This Week in Science, wherever podcasts are found. Plug us into your ears, if you will. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up while you're there for our newsletter.
You can contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistmeanin@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered all the way into the mycelium of a giant honey mushroom.
ooh, that would be awesome.
That sounds like fun. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world, so I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice, show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand, and all it'll cost you is a couple of grand. 'Cause this week's science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way, so everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Oh, Sadie loves snacks.
Yes, she does. Hot Rod, how dare you? She is not chunky.
That is, yes. Don't comment on that stuff.
That's, first of all, I body score her daily.
Do you?
I was a zookeeper, and I know what to look for. Could she lose like a pound or two? Probably. But also, corgis are
chonks.
And so she's not a chonk. She doesn't, her tummy's not
She's
like hanging down. She still has a whoop, behind her rib cage, which is good. She still goes in, if you look down from above, goes in behind her rib cage, which is what you want. So she's okay. She's fine. But right now we're not running, so she actually might get fat since we're not running right now. We'll see. I had to take a break because we were moving.
Yeah.
But then I on my first run in my new neighborhood, I tripped on a root that was poking through the cement and just ate it in the worst way possible.
Ow.
Yeah.
Are you okay? You're not okay.
I think I'll be okay. I'm hobbling around with a cane right now, but
Oh.
but I can bend it, and I can gingerly put weight on my leg. It's just I think it really needs to heal.
Did you break your patella?
I don't think so. I don't think anything's broken. I feel like I'd be way worse off if anything was broken.
Yeah.
I feel like I just hit it. So it was just pure brute force on my kneecap.
Ow.
My entire body weight pretty much went on my kneecap.
Ow. That sounds terrible.
Yeah, on cement. So, and I was trying to dodge her because I didn't want to fall onto her.
Don't fall on the dog.
So, yeah. Anyway, so we won't be running for a while.
I'm sorry,
but that's okay. That's the thing is as you get older, the bounce back for injuring yourself gets longer and longer.
It does. Let me tell you.
I'm sorry, and I'm already experiencing it. So definitely.
Oh my goodness.
Just a fall is a thing now that can change your life. How crazy! Just not putting your feet in front of each other correctly change everything.
Mm-hmm. Yeah. Well, I mean, at least it'll change your perspective. You'll be like, "Hey, how'd I get down here? World looks different."
Well,
on the ground.
Hang on a sec. So what did I miss the explanation of what the injury seems to be?
Fell.
You fell.
I wanged my knee.
So
technical term.
Yeah.
Do you think it's a little muscle tear? One of those.
It's I don't know.
ACLs
I mean,
or
I don't know a lot about knees.
Okay. Well, okay. So this is my son tore his ACL. This is one of those.
Oh, jeez.
Really bad in a weird freak accident where he was actually at a dog park. Dogs are always responsible for these.
Okay. All right, you dog hater.
He doesn't like animals.
But he was running and a dog ran in front of him, and so he stopped to not run into the dog, and somehow that interaction
Twisted.
tore that muscle.
Wow.
He was in a leg brace for months and months. There was talk about maybe he might need a surgery. He was young enough to be able to restore it without. But it's very, small muscles that are involved in this. This is one of those things, perfect example of technology we've gotten from sports.
Mm-hmm. Yeah.
It is a very common sports injury. And therefore, there have been so many procedures done over the years that it's almost a routine thing now if it came down to a surgery. I'm not trying to set you up for surgery, but I am telling you need to go get it looked at really soon.
Yeah, it's tough because I can't drive myself to a doctor right now. So there's that because it's my right leg. Yeah. And also I have a new job, so I don't really have any sick time. So there's that. So that's
I'm not understanding any of what you're saying. So you're saying you're going to put off what could potentially be a catastrophic injury that lasts your entire life for some of these short-term. Have you heard of Uber? Is that still a thing in COVID?
It's COVID, man.
Did they shut it down? I don't know. I don't leave the house
Uber's
anymore.
so gross during
You
COVID.
can have the windows open.
Yeah, but anyway,
all the windows open.
No, I'll get it taken care of. I'm giving it a couple days so that I actually know what's wrong with it a little bit better. Because if I went to a doctor yesterday, I'd just be like, everything hurts. So I'm elevating it, I'm icing it, I'm working from home. So luckily,
sometimes it's just a bad fall.
Much. Yeah, and it could just be a bad fall. Like yesterday I couldn't stand at all, and today I can hobble around the house. So I'm already healing a lot faster than I would think if it was something bad. But yes, if I am still in pain in a few days, I am going to go to a doctor.
Good.
Okay, good.
Yes. No. I'm not going to have irreparable damage to my knee. I need it.
Don't do irreparable.
Yeah.
Reparable. We want.
Yes. The knee's important.
Yes.
Yeah.
Yes.
So no. I'm definitely gonna.
No playing. Yeah. Just you want to be able to play soccer on a sandy beach for as long as you want.
Soccer's gross.
My hair is not.
Wait, why is soccer gross?
Because I played it as a child and I didn't have very much fun.
Okay.
I was a casualty of the Viking soccer league of the 90s.
Okay. But, soccer, by the way, is one of those sports that America, the female soccer players excel at that sport better than we excel at most international sports. They're like the best team ever. Just saying. You might want to take a second look at your soccer is gross comment.
Well, I don't think we should force small children to play soccer en masse.
You know what's funny? That because America, doesn't have a lot of professional or well-publicized soccer leagues. It's like everybody the soccer moms. It's the thing. Everybody's taking kids to play soccer, and then they just stop when they get to college, apparently. Because we never see the televised. It just becomes an unpopular sport in this country.
It's because you force children to play it, and then they're sick of it. That's why.
Yeah, but you would think, with the amount of emphasis there is on youth soccer, we would have college games being played, like we have NBA college NCAA, football and basketball, college athletics, but you don't see soccer represented there on that level, even though I think more people play that as a youth than any other sport.
And I'll tell you, that's why. Everyone's had enough.
People love soccer in the Pacific Northwest.
Oh, good. Yeah,
it's huge up here.
It's huge here, too. It's huge everywhere.
It's huge. Huge youth soccer. Huge. So Blair was saying that she wanted, because she's in pain,
Hey, Blair.
not to stay
Blair,
and hang out all night
I'm
long.
kind of getting tired of hearing you complain
but
about
one
your physical.
But one moment before we dive through.
Set me up to set up the. Okay,
I was, but I was just setting up in a kind of slow, long, slow way. But I was thinking tomorrow I'm going to be working and doing some stuff, but I was thinking of throwing on, if you're going to be watching the NASA lander, well, I mean, can't watch it land, but watching some of the coverage to make sure it is landing and that they get signals back and all that kind of stuff.
Watch the crew go like,
yes, we can watch.
It would be really funny if we
watch that.
We got footage from Mars of it landing, and we all watched it, and then afterwards went, wait a minute.
What? I mean, they've got Mars orbiters and things that have cameras. Just move those cameras for a second. Watch it go in. Anyway, I was thinking of doing a little, just doing a Twitch stream to chat with people, other people. I think it's at 11:15 a.m. Pacific time.
That works.
Is when coverage starts, and then 12:15 they're supposed to get confirmation of it hitting the atmosphere of Mars. Okay. And so then it'll be somewhere. It's somewhere between the noon and 1 o'clock hour.
Just tell me when to show up.
Okay, I'll text. Should I text you?
Yeah, just text me when to show up.
Send me a group text.
I'll send you an invitation. And we'll do a StreamYardy thing, and we can hang out and talk and watch the NASA coverage and be like, dude, what? And stream to Twitch, and it'll be fun.
Although the last NASA thing I watched live was the Challenger. I'm just saying I might be bad luck.
There is just you might be bad luck.
I'm 1 for 1. That's all I'm saying.
1 point it's just a point. It's not even a line yet.
I understand. I'm still 1 for 1. That's all I'm saying.
You're also 1 for 1 on predicting the Super Bowl.
Oh yeah.
Oh no, that was fantastic. No, I made a lot of other predictions that didn't. But if you recall that prediction though, that was something like even frighteningly.
I actually just looked at it because it was 10 years ago.
Okay.
It was 10 years ago last week.
New Orleans Saints versus the Indianapolis Colts.
Yeah, and you
I didn't just predict who won.
You predicted the point spread. And
the final play, I was like, Peyton Manning, who doesn't throw interceptions, throws an interception. Oh no, it's taking it back. Oh, pick six. Oh, and that's what happened. And it was really scary to watch the game unfold the way you predicted it.
Did you even remember?
No, I totally did, because I was watching the game.
And what is going on?
And somebody in my Twitter feed or the Facebook feed, because I used to watch those things once upon a time, was freaking out because they knew the prediction and just watched it happen. So yeah, then the next year I sold sports pick'em book picks for the first, I think, 3 weeks, but I got like everything wrong, so and it ended that.
Yeah, that ended. Like I said, it was 1 for 1 for a hot second.
Yeah, it's actually this is an ancient scam where you would use mass mail to predict the outcome of a sports game. And so you send different picks, sometimes they conflict, to 1,000 people. And at some point you've got a subpopulation there where you've been right every week. It's like picking every box in one of those what the scores are at the thing. But you send it to 1,000 people. And after the first week, maybe you're down to 500 people. Maybe after the second week, you're down to 250. Week 3, week 4, you're down to 50 people. But to those 50 people, you've been right every time. So now you're like, $50, I'll give you the next answer. Then everybody sends you $50. Half of those, maybe a third of those, quarter of those, you get 3 or 4 games right. those people are going to be down for $100 next time because they bet on the game with your $50 and they won.
I like your math. I like your math here.
It's the greatest, like, it's one of my favorite scams of all time because somebody at some point figured out, like, yeah, we can predict all of the different outcomes.
Use math to make money, honestly, or I could use math to scheme.
Well, that's what, okay, yeah. Well, now, to be fair, when it comes to Wall Street, is there a difference?
Yeah, no, that's scheming for sure. Yeah.
Hey, Blair, I wish you a speedy recovery.
Thank you.
Go ice that knee.
Yep.
And say goodnight, Blair.
It's right here. Good night, Blair. Say goodnight, Justin.
Good night, Justin.
Good night,
Kiki.
Good night, everyone. Thank you for another great show. And hey, maybe we'll see you tomorrow to watch the NASA stuff. But for sure, we'll see you next Wednesday because we'll be back. And I hope that you are too. Have a wonderful week. Bye.